everyone, my name is Sonam Yangchen Sherpa. I'm from grade 9 Gandaki. Today I'm going to perform an activity, extraction of crude DNA. DNA is a molecule that contains the genetic instructions for the development, growth and reproduction of all organisms. DNA extraction is the process where scientists take out the genetic material DNA from the cells. It allows them to study the DNA and use it for different purposes like medical research and solving crimes. DNA extraction helps scientists to study the genes and understand how traits are passed from parents to offspring. It is also crucial in medical research, helping scientists find treatments and cures for genetic diseases like cancer. Now let's perform the activity. For the sample, we have taken strawberry. Now we'll put the strawberry in the Ziploc bag and crush it. By crushing the strawberry, the several of the cells will be crushed. Let us make the reagent that will break the cell membrane and the nuclear membrane. First we'll add the water. And let's add some detergent. We'll also add some salt. And we'll mix it. Now let's pour the reagent into the Ziploc bag. Now let's give it a gentle press. The detergent will break through the cell membrane and nuclear membrane, while the salt will stabilize the negatively charged DNA. Now let us strain the solution from the mixture. Now to this extract, let us put some isopropyl alcohol. This stringy precipitate is the crude DNA extracted from the sample. For more educational videos like this, please like the video and subscribe the channel.